Hello friends. So today we will cover the topic of genetic engineering of course code RBT 501 unit 1 and myself Dr. Deepak Gola, assistant professor in the Department of Biotechnology in Noida Institute of Technology. So the first uh, unit include the basic concept of genetic engineering and enzymes, the libraries, genomic libraries. So today we will learn about the basic concept of gene cloning and the selection of the recombinant. So in this, the first, what is gene cloning? So first we have to learn about the clone. What are clones? Clones is a Greek word which means twig. It's the meaning in English. It means the exact copy. So in this we have to create the exact copy. If someone say that it is the clone of like a plant, it means these are genetically as well as phenotypically the copy of each other. So the next as per the definition, it is the production of the exact copy of a particular gene or a DNA sequence using genetic engineering techniques. So in this we produce the gene basically. What is gene? As we know, gene carries some hereditary information like the information about your height, color of eyes, color of skin. So we have to make the clones of those genes. So for this we use gene cloning techniques. In this we will isolate that gene, and after isolation we will use some enzymes called restriction enzymes, and we will use cloning vector to insert that enzyme into the cloning vector, and this vector now called the recombinant vector, and after that this cloning vector we will insert into the host cell. Host cell is what it will carry that vector, and it will in that our vector will multiply and make the number of copies of that gene. So inside the host cell, the recombinant DNA undergo replication. Thus a host will give rise to a colony of cell containing a clone target gene. So next, what is the minimum required to produce a gene cloning? So first of all, we require a gene of interest. So it can be like gene of insulin for diabetes, it can be gene for height, it can be gene for your color, skin color or hair color, eye color. The next is vector. What is vector? Vectors are the vehicle. If we want to go somewhere, use automobiles as a vector to move from one place to another place. Here also we require a vector to move gene from one place to another place. The next is enzymes. So there are basically multiple type of enzymes. Here we require is the first category is the restriction enzyme, and next is the ligase enzymes. So we will learn about in the later slides. We will learn about these enzymes, how they work. And next is the host. Host is where we insert our vector. In host, the vector will multiply, make the number of exact multiple copies of our gene. And the next is the media for selection. So first is this is the basic concept. Here we can see we have the vector which act as like it can be a plasmid, phasmid, cosmid, and in this vector we will insert our gene of interest. So we use the restriction enzyme for the same. We will learn about these restriction enzymes and the vector in later slide. This is the basic concept. So the target we have to insert this target gene into the plasmid. For this we use the restriction enzyme and the ligase enzymes. Ligase enzyme, as we know, we have learned in the DNA replication, it combine the two DNA fragment or the primer or basis oligonucleotide together. So after ligation we will the plasmid that now will call the recombinant plasmid, and we will insert this plasmid into a particular host, and the process is called transformation. So here is the proper definition of the vector in term of genetic engineering. It is a DNA molecule which carry a exogenous DNA into a host organism and facilitate stable integration and replication inside the host system is termed as a vector. So for to be a vector, there are some properties which should be there in a vector. First of all, it is capable of it should have a self replicating capability. Like in plasmid, we know plasmid is a circular DNA, extra chromosomal circular DNA, which we found in a bacteria. So what plasmid did, it provides some resistant properties, it help in fertilizations. Plasmid can replicate itself. Next, it should have a restriction site. Restriction sites means we have to cut our DNA, our vector and the DNA with the same restriction enzyme. So the in the fragment which we want to replicate, the gene of interest we want to replicate, we have to insert that into a restriction site available in the vector. Thereafter, it should have a origin of replication, so it can replicate itself. Then it should be should small in size, but can carry a large amount of insert, like large size of insert. It should be, why it should be small in size? Because if it's large inside, we cannot insert that into a particular host cell like bacteria, because it can bacteria also is capable of like something, it cannot beyond the some other size, it cannot take that plasmid. So after that, it should have a selectable marker, because after like in genetic engineering, it is when we are preparing for gene clone or recombinant plasmid, it is 50% chance that our recombinant like gene of interest is inserted into a plasmid or not. So for that, we should have a selectable marker. What selectable marker will do? It will help us to identify that the host which are carrying only recombinant gene, recombinant plasmid, not the one which have intact plasmid. So these are the different type of bacteria we have, like vectors we have. The first is the plasmid, which is source is bacteria, and what is the size here? It can carry only size of 15 kb, less than 15 kb. And what is it used for? Normal gene cloning and complementary DNA library. We have to our expression assay. The next is the phase. What are phase? Phase are the viruses, as we know, which can infect like bacteria. Here we have a bacteriophase, and it have a we can insert gene of interest, can be a size of 5 to 20 kb. In that we can insert only in between 5 to 20 kb of gene of interest in this, and this is used for preparing a genomic DNA library, then complementary DNA library and expression library. Next is the cosmid. Cosmid is a recombinant molecule which contain the cos sites from the bacteriophage and plasmid. So it's cosmid. Mid is from the plasmid and cos is from cos site. So it can have the insert of around like 15 to 45 kb, and it is basically used for preparing a genomic DNA library. Next is the BAC. BAC is bacterial artificial chromosome, and it can have a large size 75 to 300 kb, and it is used to analyze the large genome size like for human. Next is YAC. YAC is yeast artificial chromosome, and it can carry up to 1 MB insert size, and it is basically found in Saccharomyces cerevisiae. Contain the centromere region, telomere region, and autonomous replicating sequence from the yeast Saccharomyces cerevisiae, and the application is the analysis of the large genome. Next is MAC, that is mammalian artificial chromosome, which contain the insert of like between 100 to 1 MB, 100 kb to 1 MB, and it is basically used in animal biotechnology and human gene therapy. So first of all, we will learn about the restriction enzymes. What are restriction enzymes? These are basically a restriction nuclease. What are restriction nuclease? These are the enzymes which can cut down the DNA or RNA molecule. So restriction enzyme is a nuclease enzyme that cleave the DNA sequence at a random or specific site known as restriction sites. So for these are also called molecular scissors, because these are very small in size and they are cutting the DNA. So these are like they are cutting the molecule, so they are also called molecular scissors. So there are two type of nuclease enzyme. The first is exonuclease and second is endonuclease. Exonuclease, as we know, So, in this we will explain the exonuclease and the endonuclease. We can see the what exonuclease do. It will we know the DNA have 5' end, 3' end, and then this is 3' end and 5' end. So, exonuclease will cut only from the ends. So, it will cut only from these ends, whereas in case of endonuclease it can cleave from anywhere inside the in between the strand, like this, not from the ends. But in case of this exonuclease, they will cut the DNA from ends only. So, this is the basic difference from endonuclease and exonuclease, but the what is the common, that they will break only for cleavage the DNA molecule. For cutting the DNA molecule, they will only break the phosphodiester bond, which is the backbone of the DNA molecule. So, automatically if the backbone is not there, though the DNA molecule will cut down into small pieces. So, these are the 3 type of restriction enzymes which we found in the bacteria. So, what is the basic use of restriction enzyme? These are the enzymes which help the bacteria to differentiate, to cut down the DNA molecule or the virus molecule which are attacking them. So, these are the different type of restriction enzymes: type 1 RE3, type 2 and type 3 restriction enzymes. So, on the basis of their different properties like abundance, recognition sites, nucleus, subunit structure, cofactors, thereafter DNA cleavage requirement, site of methylation, they are differentiated into 3 categories. The first one is the least common, then the second one, and it cut the restriction site. It recognize the DNA from one site, and after recognizing the DNA site, it cut down the DNA after 1000 base pair, whereas in type second it recognize and cut down the DNA molecule from at the same place. And here in this, the cleavage of it's recognize the strand 20 at one place, and after 24 to 26 base pair it cut down the DNA. So, these are the not as like heterotrimer, it is made up of 3, basically 3 subunits of different shape and size. Type second is homodimer, it is made up of 2 subunit of same type, and the third is heterodimer, 2 subunit of different shape and size. These are the cofactor like ATP, Mg2 plus, SAM is S-adenosyl methionine, and these are magnesium, and in this case of magnesium and SAM. And in this DNA cleavage, what is the requirement for cutting the site? To recognize site in any orientation. In this case is only single restriction sites is required, and in next to recognize site in head to head orientation. Next is the site of methylation at recognition site, at in this case also recognition site, and in last also recognition sites. So, what is the need of these enzymes? So, let us see if there is a, this is some like E. coli we have, and some virus is attacking. So, if there is a DNA of virus and we have a DNA of, this is for virus DNA, and this is the E. coli DNA, and there is a restriction enzymes here. So, how will it recognize that it won't cut its own DNA? So, for this, what this E. coli will do, it will methylate with the help of methylase enzyme, it will methylate its own DNA. So, it will not recognize its own DNA, it will cut down only the virus DNA because it is not methylated. And in most of the cases we use only restriction, generally in like 99% cases we use only restriction 2 type enzyme. And why so? So, we can see like if we have restriction 1 enzyme, and there is an enzyme, it recognize the site here. This is enzyme, restriction enzyme. So, it will recognize here and it will cut down the like after 1000 base pair. So, it will increase the size of our insert, but in case of restriction site enzyme type 2, it will recognize the site and it will cut down the site from the same place, where in case of restriction enzyme third, what will happen, it recognize the site here and it will cut down after 24 to 26 base pair. So, it will also increase the size of our insert, because for this we will require first of all we require a recognition site, and there is also a cutting site which is 1000 base pair. So, automatically it will increase the size, and it will increase the size of our insert by 24 to 26 base pair. Hence, we used restriction enzyme because it will recognize and cut down at the same place. So, next enzyme which we will use is a DNA ligase. As we know, we have in genetic engineering we have enzyme called DNA ligase, which can bind the two DNA molecule. If these are two DNA molecule, in replication also in the previous class we have learned that what DNA ligase, it will join ligate this DNA fragment into a single piece if they are fragment piece. So, how it will do, these are the sticky ends which are there, which are produced due to the restriction cutting sites, and it will bind with the help of these, because these base pair are A is bind with is complemented to T, A is complemented to T, and T is complemented to A, and this A is complemented to this T is complemented to this A. So, it will this two fragment will bind together and produce a one fragment. It can be a recombinant fragment or any type of DNA fragment. So, these are the basic concept. So, these are the vector, we have learned that these are the vector plasmid. They are different. We can use another type of vector like BAC, YAC or phasmid, cosmid. Then we have the target gene. We will use the restriction enzyme second only. Thereafter, we will join our plasmid with the target DNA with the help of ligase enzyme, and it can join two DNA fragment together and it form a recombinant plasmid. After that, this recombinant plasmid we will insert it into the host. So, this process of inserting the recombinant plasmid into a host is called transformation, and after that we have to make the selection that how we will recognize, because it can be a possibility like it can be a intact plasmid also, So, we have to make the selection. So, possibility is that it can be a 50%, it can be a 50%. So, possibility is there that it can also contain the intact plasmid or the recombinant plasmid. So, we have to make the selection. So, for this is selection on the basis of their antibiotic resistant sites. So, as we can see, we have the restriction site for the enzyme PST1, and thereafter we have a two antibiotic resistant site here. First is ampicillin and next is tetracycline. It is tetracycline resistance and ampicillin resistance. That means that if a bacteria containing this plasmid will able to grow into a medium containing antibiotic known as ampicillin as well as tetracycline. So, what we will do, we will the one restriction site is available in the ampicillin coding gene. So, what we will do, we will cut down that ampicillin gene and we will insert our gene into the ampicillin coding site. So, what it will do, it will mutate this, it will mutate this gene So it will not code for the protein which will help the bacteria to grow into the ampicillin medium. So we will cut down the DNA, we will insert our DNA at this place with the help of DNA ligase. We will combine these two molecules, and after this we will transform this into a particular host. So host can be like E. coli or some other bacteria. So this is like foreign DNA which has been cut with the PST is inserted into the ampicillin gene, and thereafter we will transform this into a tetracycline grown into a media containing tetracycline. So we will learn ki how will it will do. Like, let us see, this is our plasmid, and okay. This is ampicillin resistance, and this is tetracycline resistance. So what we will do, we will disrupt this gene, and we will insert of our gene of interest. So we can have like gene of interest of this color. So we will insert this in here. So now how it look like after insertion. So this is the plasmid, and this is our gene of interest here, and here we have tetracycline gene. So this is gene of interest, GOI. We can say it is GOI. So after this, what we will do, we will insert into the host. We will insert into the host. Host can be like E. coli. It can be E. coli. So it is a possibility that gene of interest is there or is not there. So there are two possibilities. So in this we can have a intact plasmid, and this we have a recombinant plasmid. So this can grow, cannot grow, this cannot grow into ampicillin containing medium, and this can grow into both, and it can grow in tetracycline medium, but it can grow both, it can grow in ampicillin medium also and tetracycline also medium also. So what we will do, we will put our host cell, this is host cell, we will put this into a medium, we will put this into the medium containing the tetracycline medium, and we will plate these into our Petri plates. So here we will find there are some Here we will find different colonies, and these colonies can be of this intact plasmid also. Because why? Because it can grow in the tetracycline medium, and it can be of a recombinant plasmid. Why? Because it can also grow in the tetracycline medium. So, how we will identify? We will take a velvet, sterilized velvet, and uh, uh, this is like this. This is the velvet. We will use the replica plate method, and we will put velvet in this plate. The plate I have shown in the earlier slide. We will put this plate, and we will plate these colony will attach to these sites. As we can see here, it can be seen. So, after that we will plate these colonies into a medium containing ampicillin medium, ampicillin containing medium. So, here only the one which contain intact ampicillin will grow, and other will not grow. The one which will not grow is the host which contain the recombinant plasmid. So, what we have to do, we will go back to our original plate, and we have to mark which those colonies which are growing in the original plate, but not growing in the ampicillin containing plate. So, what we will do, we will take those colonies which are not growing in the ampicillin plate, but growing in the tetracycline plate. So, we have to back calculate our colonies, and we can see here these are the colonies which are not growing. So, we will take these colonies, and these are what? The host cell which are containing what? Recombinant plasmid, which are containing the recombinant plasmid. So, this is how we screen our host cell which contain the recombinant plasmid. And as we can see, these are the ampicillin, and the tetracycline help us in the selection of the recombinant plasmid. Hence, these are also called the selectable marker. These are also called a selectable marker. So, these are the selectable markers, and in this we can say these are the plasmid, and plasmid should have some property like origin of replication, as I have told you earlier. Origin of replication, self replicating capability, it can multiply into the host. So, this is all how we select our recombinant colonies, and there are another method like blue white screening that we will discuss in the another class. So, student, today we learn about the basic concept of gene cloning and one of the method of the selection. So, that is all for the today. Thank you, and if you have any query, you can mail me on my contact ID deepakgola@gmail.com. Thank you very much.